I like decided to change my talk last minute last night, which was probably not the best idea, but I thought I would, given kind of some of the conversations I had with students yesterday, thought it would be fun to talk about kind of some two very different projects going on. So as you all know, evolution can occur in over just a small number of generations in nature. So here I'm showing you kind of the textbook examples of rapid evolution. So there was rapid evolution of melanism and peppered moths in the UK, and also kind of rapid evolution of beak size and shape in Darwin's finches. We also have seen kind of rapid phenotypic evolution caused by anthropogenic changes, such as hunting-induced evolution of decreased body size in bighorn sheep. And in some of these cases, we have some understanding of the genes underlying the rapid phenotypic change that we observe in nature. But for the most part, I would say we still lack a relatively deep understanding of how short-term evolutionary change occurs at the level of the genome. And understanding kind of the genomic basis of short-term evolutionary change is really important if we want to be able to, assess adaptive potential of natural populations and for predicting evolutionary trajectories. So as you all know, since this is the PopGen school, the field of population genetics, right, is focused on trying to understand the different evolutionary processes that underlie evolution, so that change allele frequencies in populations over time. And typically, for most studies, the way this works is you go out, you sample your population of interest, genotype a bunch of individuals, and then use computational methods to try to make inferences about the evolutionary processes that generated the pattern of genetic variation that you observe in your sample. If you're lucky, you may have many different temporal samples from the same population over time, which gives you a lot more power to make inferences, especially over kind of processes occurring over shorter timescales. But one thing to keep in mind is that what is really happening in natural populations is that different individuals are moving around the landscape. Some individuals survive, others die, some individuals reproduce. And essentially what this means is that different individuals make different genetic contributions to a population over time. And it's this variation in individual genetic contributions that underlies allele frequency change. So these individual level processes are fully encapsulated in what's called the population pedigree, or the relationships of all individuals in a population over time. So for every natural population, there is one true underlying pedigree. And if you have information on this pedigree, then you have the ability to directly observe the evolutionary processes changing allele frequencies over time. So this is a lot of work to be able to get information on the population pedigree, but there are a few cases where these type of data are available. So these mostly come from long-term demographic studies where scientists have studied individually marked animals in a single population across many decades. So right now, kind of the most complete population pedigree comes from these individual-based studies, or if you're in the lab, from experimental evolution studies. But one of the things for natural populations is we've gained a lot of insights from these handful of long-term demographic studies. And by overlaying kind of genomic data to the extensive demographic data, we can answer a whole bunch of other questions. So my lab is really interested in kind of doing population genetics with pedigrees. So we develop genomic resources and tools to combine evolutionary genomics with these multi-decade demographic studies that have extensive information on individual phenotypes and lifespans and kind of natural history and ecology, in order to answer a lot of questions about short-term evolution in natural populations. So broadly, we're really interested in understanding kind of what are the evolutionary processes that govern patterns of genetic variation over space and time. What is the link between individual genotypes to phenotypes to fitness? And eventually, what is the impact on kind of population trajectories? So a lot of the work done in the lab is with this beautiful bird, the Florida scrub jay. So this bird is a cooperative breeder, which means that offspring often delay dispersal and stay around at home to help parents raise future generations. It's restricted to this really unique fire-maintained scrub habitat. You can kind of see what the habitat looks like in the photo. It is restricted to the state of Florida and the southeastern United States. Uh, there's been a lot of habitat destruction over time, so it is federally threatened, and a lot of our work has kind of conservation goals and implications. And there are a few aspects about the biology of the study species that makes it very amenable to long-term field studies. So it's non-migratory, it's highly philopatric and territorial. So individuals tend to stay in the same geographic area for their entire life, and their offspring don't disperse very far. So we can track basically multiple generations over time. They're socially and mostly genetically monogamous. There is a very, very low rate of extra-pair paternity, but for the most part, we can infer kind of or reconstruct accurate pedigrees from field observations alone. Another aspect that makes it really important for long-term field studies is these birds are addicted to peanuts, which makes it pretty easy to work with them in the field. I will say we try to uh, interact with them as little as possible, so these photos are kind of exceptions to the rule. So there's been an individual-based study at Archbold Biological Station that was started in 1969. And there's a ton of work that goes into this field study. All of the birds in our population have a unique combination of color bands on their legs, so we could identify individuals in the field. The entire population is censused once a month, providing accurate data on individual lifespans for all the individuals in our population over time. Every nest created by all the family groups we monitor is found and really carefully monitored, so we know exactly how many eggs they lay, we know how many of those eggs hatch, and we know the fates of all of the nestlings born in our population. The censuses we collect spatial data too in the censuses, so we have accurate direct measures of dispersal distance, which is nice. And then importantly for my work, my colleagues started collecting blood samples so we can extract DNA from every new nestling or incoming immigrant in the population starting in 1999. So we have this really nice archive of blood and DNA samples for all the birds in our population. And then finally, there's also a whole suite of ecological data. So we have data on habitat quality, climate measures over time, fire history, food abundance, etc. And so over the past more than 50 years, we've accumulated complete life histories and phenotypic data for more than 10,000 individuals on a 15-generation pedigree. So this mass of symbols and lines is our population pedigree. I only show it to show you we have a lot of data. I mean, not that much data, but quite a bit of data for a natural population. And then we also have fairly comprehensive genotyping of our population over time. So as a graduate student, I sequenced and assembled the genome and developed a custom Illumina iSelect B chip for about 10,000 SNPs across the genome. And on the bottom here, this histogram is showing the total number of individuals in our population over time in gray. And the blue bars indicate the individuals we have genotype data for. And so you can see we've genotyped nearly every single individual in our population for more than about 10 years. What we're doing right now is going back and doing whole genome sequencing of all the birds in our population from 1999 to 2021, but unfortunately I don't have, those data aren't ready yet, so I don't have stories to tell from there. But this wealth of genomic, environmental, demographic, and phenotypic data from a population with a known genealogy or known pedigree provides a really powerful framework for directly testing a lot of different predictions of evolutionary biology in nature. So today I thought I would tell you two totally different stories. So first I'll talk about some work we're doing trying to understand the genetic architecture of fitness and looking at what maintains genetic variation in our population. This work is done in collaboration with Alyssa Cosgrove and Andy Clark. And then I'll talk about kind of a whole suite of different projects focused on understanding the consequences of gene flow into our kind of focal population. And I'll be presenting work from graduate students Jeremy and Rose. Okay, so let's start with thinking about genetic variation for fitness. So we now know that there is kind of considerable genetic variation for fitness in natural populations. This is a figure from a relatively recent science paper that estimated additive genetic variance in lifetime breeding success for several different mammal populations. So this kind of leads to this classic long-standing question in evolutionary biology, right? So selection should act to remove variation over time. So why do we still see so much genetic variation for fitness and fitness-related traits? So what maintains variation in fitness in nature? There's been a ton of work on this for decades, right? And overall, there have been kind of two evolutionary hypotheses. So one is mutation selection balance. So variation is maintained at an equilibrium because recurrent mutation continually introduces new variation that then is removed by selection. Or there can be balancing selection. So genetic variation may be maintained solely by natural selection. Balancing selection can take a whole many different variety of forms. So there could be heterozygote advantage where individuals heterozygous at a given genotype or have higher fitness than either homozygote. There could be negative frequency dependent selection operating in a population, or there can be antagonistic selection, right, where the same alleles have opposing fitness effects, either in different sexes or at different life stages. So we wanted to kind of look at kind of how much genetic variation is there in fitness in our study population, and can we kind of try to understand what are some of these potential forces that may be maintaining variation. So from our demographic data, we know that there's fairly high variation in fitness. Here I'm showing lifetime reproductive success, or the total number of nestlings produced in an individual's lifetime for about 1,000 individuals who've completed their lifetimes, right? So you can see most individuals don't successfully produce any offspring, but then some produce more than 40. One of the fun things about having a population pedigree is we can go beyond this like single generation measure of fitness and actually pull out all of the descendants of a given individual in our population over time. So just as an example, here's one male who showed up in our population in 1995. He had 4 offspring. None of his kids survived to reproduce, so this is the pedigree of all of his descendants in the population. And you can contrast this with another male who had 41 offspring, and his offspring were way more successful. And you can see he has a dramatically larger kind of genetic contribution to the population over time. And it's kind of fun. We can do simulations and estimate like 25% of the birth cohort in our population was descended from one of these like highly fecund males. So a lot of our work is looking at these like multi-generational estimates of fitness, but for today I'll just focus on lifetime reproductive success defined as number of offspring. So we ran a lot of animal models. So these are mixed effect models to try to estimate how much additive genetic variance we see. And on the top row, we're looking at lifetime reproductive success of all individuals in our population, and on the bottom, just for the individuals who survived and successfully became a breeder. And we ran analyses for males and females separately. And the purple kind of histogram just shows you the posterior distribution for additive genetic variance. And we find kind of, interestingly enough, non-zero additive genetic variance for lifetime reproductive success in females, but not in males. This is kind of opposite, I think, in pattern to many other studies where they typically find heritable fitness in males, but not females. But here we are. This is what we found. So we were inspired by kind of old work on selection component analyses that points out that fitness is really a complex trait and has many different many different components. So this idea was first raised by Tim Prout in the 60s, and then Freddie Christensen and Owen Freidenberg in the 70s came up with this really kind of elegant hierarchical series of tests to specifically look at selection, I guess, operating on different life cycle stages. This is called selection component analysis. We did perform this analysis on our with our data, but today I'm going to show you a different approach. So we realized, so the selection component analysis basically is testing for selection by looking at differences in genotype frequencies between individuals in different life stages. But in nature, we know that environment can play a really large role, and so we wanted to be able to somehow control for environmental variation. But we kind of like this idea of doing a really careful dissection of different fitness components. So that's what we then set out to do. Here I'm showing you the typical life cycle of a sexually reproducing diploid organism using scrub jays as an example. So we know that you can think about many different fitness components. So for instance, zygotes, there may be differential survival or viability selection. So survival from zygotes to adults. Not all adults, not all individuals who survive to adults actually successfully reproduce. So there's opportunities for sexual selection. Individuals may vary in the number of eggs they produce or fecundity. There could be variation in fecundity, and also there is an opportunity for gametic selection, but we don't consider that here. So what we did was to perform several different genome-wide association studies where we wanted to map these different fitness components. So we looked at variation in survival, conditional on surviving to a given age. Did a given individual attain breeding status or not? That was what we used to look at sexual selection. And we also looked at variation in clutch size. We included the full kinship matrix and also natal year and natal nest as random effects in our models. And then kind of the beauty and curse of having a lot of data is we had a whole suite of potential fixed effects that we needed to consider, right? So we know that many different things may affect fitness in our population, including attributes of the individual. So inbreeding coefficient, hatch date, whether or not they're an immigrant. We also looked at attributes of the natal nest. So how many helpers were at the nest, what was the territory size and fire history, and also the age and experience of its parents. And then we also had a bunch of climatic factors that varied over time. So population density, rainfall, temperature, food abundance, etc. So we did a series of variable selection approach to identify which of these factors are significantly associated with each of our different fitness components, and then controlled for them while kind of fitting the effect of every SNP across the genome to do our genome-wide association study. So we ran a ton of models, and I thought I would just give you a few highlights. So first, for survival, we looked at survival to a bunch of different milestones in a scrub jay's life. So we band individuals when they're 11 days old. That's when we can start our analyses. Scrub jays typically stay in the nest until around day 18. That's when they fledge or leave the nest, and they pretend to be little pine cones until around day 30 when they're more mobile. By day 90, they're nutritionally independent from their parents. And by day 300, they're physiologically capable of breeding. And then we also look to see whether or not they actually successfully established as a breeder. So to give you a sense of kind of our sample sizes, we modeled females and males separately and did a bunch of different logistic regressions for survival to in these different time intervals. There's a lot more mortality early on in life, so only about 60% of our nestlings survive to day 30, 45% survive to day 90, and then 30% survive to about a year. And I guess my expectation going into this was that we would find absolutely no large effect alleles, because our sample sizes are like our sample sizes are about 1,000, which is not large for a natural population, but really not that large. And so if we were able to detect any significant associations, it would have to be for a relatively large effect allele. And to my surprise, we did find 2 large effect alleles for female survival from really early life survival, day 11 to day 30. The different fixed effects that we needed to control for this particular fitness component were incubation date, hatch number, inbreeding coefficient. We know there's inbreeding depression in our population. Drought index and the maximum temperature during this time period. And we found 2 SNPs, one on chromosome 11 and one on chromosome 14, that were significantly associated with survival. And it was like a surprisingly large, I realize our effect size estimates might be slightly inflated because of the Beavis effect, but still, if you look at predicted survival of females of the 3 genotypes for one of these SNPs, you can see a difference in survival of like 30%. So there's clearly some genetic component to survival in females in our population. We are annotating the genome, and but one of the things, I'm not going to say too much about what are potential genes involved because we only have 10,000 SNPs, so we're tagging relatively large regions of the genome. For fecundity, we looked at several different measures of fecundity, but I'm going to just show you results for number of eggs laid in the first clutch of the season. So mostly scrub jays lay 3 or 4 eggs, some lay 0 to 5, but for the most part, it's 3 to 4. In females, we found different fixed effects that matter for female versus male fecundity. And if you do the genome-wide association study, you can see that we found 3 SNPs that were significantly associated with egg numbers in females and 2 different SNPs in males. So overall, we found kind of 12 alleles that are significantly associated with variation in fecundity, and then 2 associated with kind of survival in females. But what we were really interested, so like we totally recognize our GWAS is a little underpowered. These are we expect that these are highly polygenic traits, right? So really we were interested in trying to see what we can learn from looking at kind of effect sizes of all of our SNPs together. So what we did was kind of a simplistic test, but we just looked at correlations between genome-wide effect sizes for every SNP with a like suggestive p-value, like a raw p-value of 0.1. So here I'll be showing you a bunch of different correlation plots for our different fitness components, so survival and our different survival intervals in pink. Yellow is our measure of sexual selection. The teal shows our different measures of fecundity, and the purple is lifetime reproductive success, either of all individuals or just breeders. So for females, you can see for the most part, effect sizes are positively correlated with each other, although we do see some fun negative correlations genome-wide. There's a similar pattern for males. And so I just wanted to point out, I know this is a lot, I just wanted to point out 2 fun patterns. One is if you look at correlations of effect sizes with lifetime reproductive success, we can see that survival is most correlated with lifetime reproductive success, which is nice and consistent with kind of life history theory, where survival is a primary driver of fitness in longer-lived individuals. And then it was fun to be able to see some evidence of life history trade-offs. So for instance, in females, alleles that are associated with longer breeder lifespans are negatively correlated with, they're also associated with lower egg num. So we kind of recover the classic life history trade-off between survival and reproduction just by looking at our correlations between the genome-wide effect sizes of our fitness component GWASs. And then if we look at differences between females and males, you can see we find a large number of negative correlations. And so there may be kind of evidence of sexual conflict kind of operating across the entire genome. So here, this is a project that we're like just wrapping up, and so I've been kind of trying to mull through the results a lot. But we did this really careful mapping of lifetime reproductive success as well as these different fitness components. Um, and it was fun to find that lifetime reproductive success is heritable and primarily driven by variation in survival. And if we look at our effect size correlations across the genome, we see evidence of trade-offs between survival and reproduction. We also found trade-offs between kind of early survival versus later survival, and also some evidence of sexual conflict across the genome, which just suggests that kind of antagonism may be playing an important role in maintaining variation in our population. One of the things I will say is that these analyses, we kind of ignore indirect genetic effects, which can be really important. So we have some ongoing work right now where, because we have family groups, we have the ability to not only estimate kind of direct genetic effects of alleles transmitted by parents to their offspring, but we can also estimate indirect genetic effects. And we have some preliminary evidence of like um, strong paternal effects on phenotypes such as nestling weight. But this is something we're still working on. I just wanted to give a preview. Okay, any questions before I switch topics? Yeah. For the most part, they form long-term pair bonds, but there are a few rare cases of divorce, and then they can also switch mates if like their first mate dies, they'll repair. Yeah. Yep. That's a great question. We are trying to disentangle that. Right now, it's a little tricky because number of mates is correlated with total breeder lifespan, right? Like if you live longer, you're more likely to outlive your mate and then repair. Yeah, so it's tricky. I think it's mostly lifespan. What we're trying to do now is to test what's the negative effect of mate switching, because we know that pair experience, so how long a given pair has been breeding together, is positively associated with kind of nestling with fecundity and nestling survival. So we're working on kind of disentangling the effect of mate switching. Yep. Okay. If there aren't, and like feel free to ask questions about the first part later, but I thought I would switch gears, which I normally don't like to do, but I thought it would be interesting to talk about kind of totally different questions that we're asking in the lab. So here we're really interested in understanding the consequences of gene flow, or mostly just dispersal between different populations. So dispersal, right, is a really important phenomenon in nature that can influence biological processes at many, many different levels, right? So dispersal is going to, at the individual level, it'll impact individual levels of inbreeding and also fitness. Across the broader landscape, dispersal underlies kind of any patterns of spatial genetic structure we see, and then between population, it'll impact metapopulation dynamics. And understanding the impact of dispersal is interesting from just basic biology reasons, but also has really important applications, especially for conservation. So we need a better understanding of genetic and fitness consequences of dispersal over both short and long timescales in order to better predict how populations will respond to habitat fragmentation that is occurring worldwide, and also to better understand the effectiveness of management strategies such as assisted translocations. So one of the hot topics in conservation right now is genetic rescue and whether or not we can use that as a conservation management tool. But we don't have a good enough understanding of the long-term consequences of dispersal, I think, to really assess the effectiveness of genetic rescue. So the problem is that studying dispersal can be quite hard in natural populations. Ideally, you would want to have direct measures of dispersal distance, which is very rare in nature. A lot of folks use population genetic inference to try to infer dispersal between populations. Ideally, you would kind of hand in hand with direct measures of dispersal distance. Ideally, you would want to be able to identify immigrants coming into your population and then be able to track individual lifetime reproductive success across multiple generations if you want to assess longer term consequences. And so long-term demographic studies are really nice because if you have detailed monitoring, you can actually identify incoming immigrants into your population over time and also track kind of how many offspring, grand offspring, et cetera, that they produce. So in our study population, from the census data, we found that there are relatively high levels of immigration coming into our population over time, and it is significantly decreasing over time. So this is just the number of new immigrants appearing in our population. The decrease in immigration is not surprising. So our study population is on this track of, it's on a field station, it's a track of really well-maintained scrub habitat. The surrounding area is not as well maintained, and so we know that the peripheral populations are a lot smaller, and many of them are declining, and some have kind of gone extinct over the past few decades. From our genomic data, we could kind of genotype everybody, and we found that immigrants were much less heterozygous compared to residents. So here I'm showing you mean genome-wide heterozygosity for breeders who were born in our population in blue, and then known immigrant breeders in yellow. And you can see that there's much lower heterozygosity of immigrants, which is consistent with our hypothesis that they're coming from these much smaller and presumably more inbred populations. However, if you look at patterns of pairwise relatedness, these incoming immigrants are important for contributing levels of genetic variation to our population. So on the bottom, I'm showing genomic relatedness measured as kind of mean pairwise identity by descent for resident breeders in blue, immigrant resident breeders in green, immigrant breeders in yellow, and then all breeding pairs in our population, or all potential male-female pairs in our population in gray. So you can see that immigrants are much less related to each other than our residents. And we can actually go a step further than this and use simulations on our population pedigree to estimate the cumulative expected genetic contribution of immigrants to our population over time. So here I'm kind of starting simulations in 1990 and then looking at the black line shows the total expected contribution of all immigrants showing up in our population starting in 1991 and moving forward. And the colored lines just show the kind of the stacked contributions of each incoming cohort. So by 2013, about 75% of the genetic variation present in our birth cohort was actually introduced via immigration. Florida scrub jays have sex-biased dispersal, so more females. We have more females dispersed farther than males, and this also means we have more female immigrants than male immigrants. And so we can actually look at the contributions of individuals, immigrants of different sexes. So here I'm showing you the contributions of females in green and males in purple. For a given autosomal locus, we see a higher contribution for female immigrants, which makes sense because there are just more female immigrants showing up in our population over time. Birds have ZW sex chromosomes, so females are the heterogametic sex, so they have one copy of the Z and a W, and males are ZZ, and they transmit their Z chromosome to all of their offspring. So we did simulations to look at contributions of female and male immigrants to the Z chromosome. And you can see that males here have a higher expected genetic contribution than females. And this is just kind of a nice indication of how sex bias, like thinking about how the sex of the immigrants may have different effects across the genome because they will have differential contribution to different regions of the genome. So I mentioned at the beginning that variation in individual genetic contributions to a population is what underlies allele frequency change. So we then wanted to ask how does gene flow influence kind of specific trajectories of different SNPs. So here I'm just showing you allele frequency trajectories of our 10,000 SNPs across the genome. We had about 200 SNPs that changed in frequency by more than 0.15 over this 15-year time period, which is a relatively short time period. And some SNPs actually change in frequency by like 0.3, which is quite a bit. And so we wanted to see if these like large allele frequency shifts were driven by selection or gene flow or drift. And to do this, we use this simulation process called gene dropping in order to generate neutral expectations, right? So if you think about a lot of PopGen neutral models assume like you model a Wright-Fisher population, right? But here we know we violate a bunch of the assumptions of a Wright-Fisher model. So we have overlapping generations and we have a lot of related individuals. And so for us, the most accurate neutral model is by actually simulating Mendelian transmission of alleles down our kind of known population pedigree. So we start with kind of observed genotypes for all of our founders. So we started simulations in 1990. We also include genotypes from incoming immigrants, and this is how these simulations kind of incorporate the effects of gene flow, and then simply simulate Mendelian transmission of these alleles down the pedigree like a million times to obtain expected allele frequency distributions in our population from year to year. And so in many cases, we can observe fairly large, so for our SNPs that we see these large allele frequency shifts. So this is one example one example SNP with the observed allele frequency in blue. If we overlay on the expected allele frequency trajectories from our gene dropping simulations, you can see that they track that change fairly accurately. And in these cases, we can go back and look at allele frequencies between our kind of starting founders and the incoming immigrants and show that, in this case, gene flow is driving this fairly rapid change in allele frequency. So we know that gene flow, right? So incoming immigrants are really important in contributing to genetic variation and can drive shifts in allele frequency. What about the consequences for fitness and population dynamics? So because there's been a decrease in immigration over time, we found a significant, though very small, increase in the mean inbreeding coefficient of our birth cohort over time. And this has actually led to a decrease in juvenile fitness over time. And so this is a fairly concerning result from conservation purpose, because our population is one of the best maintained populations of Florida scrub jays left, and our results show that kind of connectivity between our large focal population and these small peripheral populations was really important for maintaining levels of genetic variation and fitness. But we can take it a step further and actually try to see what impact it has on population dynamics. So if we estimate the population growth rate in our population over time, it fluctuates dramatically over time, but the asymptotic growth rate is essentially 0, which means that our population is demographically stable. The census population size fluctuates, but on average stays the same. And so what we did was then looked at the life cycle of the Florida scrub jay, and divided individuals into different kind of life stages. So we have juveniles who can survive to become helpers, and helpers can pair to become new pairs, and then new pairs can survive to become established pairs. We separate our breeding pairs into 2 stages because we know because of known differences in fecundity between new and established pairs. And we can also explicitly model the impacts of immigration by allowing immigrants to appear as helpers or new pairs. And so essentially from our census data in this life cycle diagram, we can construct matrix population models that connect demographic rates such as survival, probability of pairing, immigration, and fecundity to changes in population growth rates over time. And so we performed what's called a life table response experiment to actually quantify how much does variation in these different vital rates actually contribute to observed variation in population growth rates over time. And so this is showing you the percent contribution of different vital rates. The most important is variation in fecundity and breeder survival, which makes sense. But we do see kind of a small contribution of immigration to observe variation in population growth rates. Because we wanted to do a more explicit test, and so oh, sorry, this is, so immigration is as a kind of the proportion of new individuals appearing in the population is shown in purple. So immigration contributes like 12%. And then the last four columns, juvenile, helper, breeder, immigrant, is the probability of pairing. So the probability an incoming immigrant becomes a breeding pair. And so that probability of pairing is shown in dark blue, and that doesn't really impact population dynamics that much. Does that answer? Yeah, sorry. It was, yeah, so it's variation in immigration rates shown in purple, and then the blue shows probability of pairing for different individuals of different life stages and how much that explains variation in population growth rates. Okay. One of the things that is also interesting, you'll notice that kind of helpers have a negative contribution to population growth rates. And what this means is that helper survival buffers the population, so it's making population growth rates very less over time. And so this is kind of consistent with a lot of ecological theory saying that the vital rates of non-breeding individuals are expected to essentially buffer a population from environmental change. Okay, so we wanted to go a step further and actually explicitly test kind of the impact of factors like inbreeding on variation in population growth rates. And we can do this via path analyses. So what we did was we took a number of environmental factors that we thought could be really important drivers of population dynamics. So we looked at acorn abundance. Area burned is just the total area of our study site that was burned within the last 9 years. We also looked at density, population density, to try to quantify density dependence. EQSOI is the Equatorial Southern Oscillation Index, so it's our kind of big measure of just climatic variation. And then we also looked at relatedness or inbreeding coefficients, and then year to just look for any other temporal factors that we don't actually explicitly consider. So what we did was we fit generalized linear mixed models to estimate the effects of these different kind of environmental factors on our different life history traits, our different like demographic rates essentially, and then connect them to the life table response experiment in order to estimate how much does like variation in acorn abundance explain variation in population dynamics. So it turns out there's density dependence is really important. So variation in population density explains about 12% of the variation we see in population growth rates over time. And acorn abundance is fairly important. Relatedness or inbreeding is this tiny green bar. So we do find a significant effect of inbreeding on fecundity and survival, but overall it does not really impact population growth rates yet. And this is consistent with some there are some hypotheses saying that there may be a lagged impact of inbreeding on demographic change in populations, um, especially if there's like density dependent growth, right? Because if you have an inbred individual, um, if inbreeding kind of decreases individual fitness, that may lead to decreased population density, which may have a positive effect on population growth if there's a lot of negative density dependence. And so this is why you may see kind of a lag in the demographic impact of inbreeding. All of these environmental factors influence population growth rate via a specific vital rate, and we can actually quantify that. So here, it's the same graph as before, but I've colored the contribution by the vital rate through which a given environmental factor acts. So for instance, you can see acorn abundance, the bar is all pink, and that just means that acorn abundance is acting only via survival, which makes sense. Like food, really, in our population, acorns are really important for overwinter survival, and so it's contributing to differences in population dynamics via variation in survival. Population density has multiple different effects, so it impacts fecundity, survival, and immigration. And then for year, you'll notice that there's a positive contribution of immigration. So kind of temporal changes in immigration are making population growth rates more variable over time. But that's being counteracted by a negative contribution of survival. So in years with really high density, there's really low immigration and low breeder survival and reproductive success. So there's some sort of density dependence going on with immigration. And if you look at the mixed effects models, we can actually see, so this is just looking at the effect of year on our different vital rates. And we can see that the decrease in immigration in our population is being counteracted by an increase in breeder survival over time. And so this kind of remarkable stability in our population, despite decreasing immigration, increasing inbreeding, and decreasing juvenile fitness is totally being counteracted by this coinciding increase in breeder survival. Why breeder survival is increasing, we're not entirely sure. Part of it has to do with density dependence, but we're looking into other factors. But this is kind of a nice kind of really careful test of what is governing population dynamics in our population. So to summarize this part, I talked about kind of a smattering of work looking at effects of dispersal. So we know that dispersal into our focal population is really important in contributing to levels of genetic variation. We know it can drive kind of some allele frequency trajectories over time. Reduced immigration to our population over time has resulted in increasing levels of inbreeding and reduced juvenile fitness, but has yet to impact our population growth rates, mainly because this decrease in survival is being counteracted or buffered by an increase in breeder survival over time. And we think it's all, again, linked to density dependent factors. What we're doing moving forward is we want to do a lot more work explicitly looking at long-term effects of immigration. So we're in the process, we're almost done, doing whole genome sequencing of all of the individuals in our pedigree. And this will give us the ability to actually track the inheritance of haplotypes in our pedigree over time and associate that with different phenotypes for different fitness. And we also want to look more carefully at kind of fitness of different generations of immigrants. So here I'm just showing, on the right, I'm showing you our birth cohort colored by different ancestry categories. So F1 here just refers to the offspring of an immigrant. F2s are grand offspring of immigrants, and we can also classify them into back crosses with another immigrant or a back cross with a resident. Well, essentially, by looking at differences between these different categories of individuals, we can ask questions about, how much is heterosis prevalent in our population? Are immigrants bringing in deleterious or kind of beneficial alleles, and eventually get a better understanding of the long-term kind of impacts of gene flow in our population. Okay, so none of the work I talked about today would be possible without the really hard work of a giant crew of interns and staff at Archbold Biological Station who collect the field data every year. I'd also like to thank my wonderful team of collaborators and students, folks who've provided comments, and my funding sources, and thank you for listening. Happy to take questions.